Hi, good morning or good evening to everybody. I'm Chandrima Bhattacharya, a current PhD student at the Tri-Institutional Computational Biology and Medicine program, which is organized by Cornell, Weill Cornell, Rockefeller University, and Memorial Sloan Kettering Cancer Center. Today, I'll be presenting my paper on global metagenomic data from MetaSUB enables city-specific microbial fingerprinting. Microbes are usually very commonly misunderstood creatures, especially since we are in the second year of a global pandemic, which seems to be never ending. But that is mostly because we have been historically studied and focusing on pathogenic microbes, which have been affecting human in some way or the others. But we always tend to forget about the various microbes which provides us with medicinal values, as well as our favorite microbial genera, Saccharomyces species, which help make the wine and bread that we love during the conferences. When studied, microbes are very insightful. That is because they are the unseen factor contributing to major earth processes, including bioremediation and cleaning up pollutants. Even in our human body, around 50 to 90% of the cells are bacterial cells. That means that we are not even the majority vote when it comes to our bodily function. Microbes in the environment has been focused for sites like Guana's Canal, which is a Superfund site in New York and super polluted, or in unique environments like the coral reefs and the Pink Lake in Australia. Being extreme in environment, they have perplexed scientists for decades. Urban biomes, which consist of microbiome of urban society, have barely been studied till like recent year when it is gaining significance. Hence, I would like to introduce you all to MetaSUB. MetaSUB stands for Metagenomic and Meta-Design of Subway and Urban Biome. This is the largest study of ongoing into daily and en route microbiome, where we use advances in biology to capture the microbes in subway and other mass transit systems. Before I proceed, I would like to introduce you to some technical terms. Previously, studying of microbes include studying of like culture technology and using microscopy to detect novel species. In today's generation, we use high throughput sequencing along with them. Here, using high throughput sequencing, what we do is to study the genetic material of a microbe in an environment, which is known as the microbiome. All living creatures are made of like building blocks called DNA or RNA. We study this genetic material for identification of the microbiome through a process that is known as metagenomics. This is how we collect data for MetaSUB. The collection technique can be very similar to that of COVID-19, where we use swabs. And using the swab, we swipe the object of interest, like for example, the ticket counter in a subway station, or like a handrail. Then we collect it and then use sequencing and advanced state-of-the-art algorithm for detecting the microbes. We have a global city sampling day, which is the day of summer solstice, on which like most of the countries and the cities go for sampling. This is the data that we have collected, and we have like more than 25,000 samples from across the world. And like we currently have more than 100 collaborators in all the 7 continents, as we also have data from Antarctica. MetaSUB is the largest study of urban microbiome, and we have recently published our finding in Cell, which you can certainly use for reference. As I mentioned before, we look at the genetic material which is present in the environment when we study microbiome. A typical sample usually consists of like 50% unknown organisms, and amongst the rest detected, it's mostly bacteria. The review paper is very interesting, and it introduces the concept of unseen majority, or the microbes which cannot be seen by naked eye. Understanding these microbes can not only help understand changes, including environmental changes like climate changes, but also how the microbes drive degradation and have survived through the multiple decades in the fast changing world. It also provides insight into the anthropological changes, which makes it extremely important to consider microbe for studying the environment. I will now discuss about the new tool I developed for studying the microbial signature or microbial fingerprint called MetaPred. MetaPred allows multiple data normalization technique and is a multi-classifier system for prediction based on microbial signature, which we can refer to as microbial fingerprint of an area. We can observe that from this plot, which is made by MetaPred's data, that microbial signature can not only be used to determine the continent from which the sample was collected, but even the cities. With MetaPred, we can predict a city with greater than 90% accuracy. As a matter of fact, we see that proper pre-processing allow almost all classifier to respond with more than 50% accuracy. The MetaSUB data is extremely robust. I used one of the top model, the random forest classifier, and used Gaussian noise. This graph shows the robustness of our model with respect to Gaussian noise. Most microbial dataset does not have as many samples as MetaSUB, and hence Gaussian noise can be added to improve overfitting as well as accuracy and robustness. MetaSUB can be used for analyzing any microbial data. One of the top issues that we face when we use a machine learning model is that they end up acting like a black box. Hence, factor analysis to determine the discriminant and the contributing species become very difficult. For a microbial data set, we have like a few thousand species, and hence it is very important to study and understand which are the contributing and the discriminant species. I developed another tool called Cassandra, which is named after the Greek goddess. And it is basically a random forest-based technique which determines the contributors driving the machine learning algorithm. Cassandra allows users to mention the minimum accuracy for the random forest. In the MetaSUB dataset across the world, we discovered 31 species which are present in across 97% of the samples. These species, consisting of the 31 species, are called core species. A core species can be defined as a species that is found in an urban environment and especially an environment which consists of like human used for the daily transportation. Interestingly, not all the 31 species that we have identified are human commensal species. That means some of the species might not be known to be like species associated with human. This is a box plot generated from the data we get from Cassandra prediction. The box plot represents the abundance of the microbes across all the samples as predicted by Cassandra's. The list of the 50 species here are the discriminant species, the top discriminant species identified by Cassandra. Interestingly, what we find is that only 10 out of the 31 core species are there in the top 50 discriminant species in driving the prediction of cities with greater than 85% accuracy using the random forest classifier. We have already shown how microbial signature can be used for predicting the cities, and also found out the microbes associated as the discriminant factors in the species. But microbial signature can predict other factors also. So here, this is basically a heat map, which shows like what other factors can be predicted using microbial signature. This heat map represents the prediction accuracy of a random forest model for cities when they are not present in the dataset. We see population is a highly predictable with microbial signature. For example, in case we were meeting in Singapore for the KDD conference, we would be carrying the microbial signature of the city of our origin. One of our projects called Stuck on You was dedicated for capturing the microbial exchange during global conferences. I discussed like how microbial signature is an important parameter, and I would like to conclude my presentation by discussing more about MetaSUB and its objective. The MetaSUB studies extend way beyond just looking at the microbial signature. Our main objective through MetaSUB is to build a geospatial, metagenomic, and forensic map. Also, currently, we see that antibiotic resistance is of growing importance. Through MetaSUB studies, we are tracking this antibiotic resistance across the world and how it is emerging as a common threat. Another important goal is for discovering novel species and the unique novel secondary metabolites which are produced by the microbes. These secondary metabolites can be used for drug discovery. And thus, I would conclude summarizing MetaSUB and my presentation by thanking the organizers and the people associated with the project. If you have any question or would like to join us as a part in our project as a collaborator, please feel free to contact us. Thank you. Now I'll be taking questions.